Assalamu alaikum. I Dr. Muhammad Akram, Department of Eastern Medicine, Government College, University, Faisalabad. Important monosaccharides. Examples and functions. Glyceraldehyde. It is an aldotriose. It is an aldose as well as triose, 3-carbon sugar. It is used as a reference sugar. It is called a reference sugar because of two reasons. All the sugars are derived from it and all the D and L sugars are referred to it. Now, dihydroxyacetone. It is a triose. It is a ketose as well as a triose. It is produced and utilized in the breakdown of glucose through glycolysis to produce energy. It is also produced from glycerol during triglyceride degradation. Erythrose. It is an aldotetrose. It is produced during the breakdown of glucose through hexose monophosphate shunt and can be converted to glucose in vivo. Ribose. It is an aldopentose. It is distributed widely in nature. In the body, it is produced through HMP shunt. Ribose and its reduced form deoxyribose are important component of RNA, ribonucleic acid and DNA, deoxyribonucleic acid respectively. Ribulose, xylose and xylulose. These are ketopentoses. They are produced through HMP shunt and can be converted to glucose in vivo. Glucose or grape sugar. It is an aldohexose. It is an aldose and a hexose. It is also called dextrose due to dextrorotations of plane polarized light. It is the main sugar in the human body. Sucrose, table sugar, is the most common source of glucose. It is the first line of nutrition for the production of energy through glycolysis and citric acid cycle. In order to get metabolized to produce energy, glucose needs to enter the cells. This entry into the cells requires insulin except erythrocytes, hepatocytes, brain cells, and intestinal mucosal cells, which do not require insulin for glucose entry. Therefore, proper care should be taken when glucose is given to diabetic patients who are deficient in insulin. Among all the carbohydrates, brain utilizes only glucose for its energy requirement for normal and efficient functioning of the brain and the body. It is therefore very important to maintain normal blood glucose level. Low level of glucose may result in hypoglycemia, hypoglycemic coma, and brain damage. Normal blood glucose level is fasting 82. Fasting should not be more than 126 and random should not be greater than 200. If glucose is in excess, it is converted to glycogen, which is stored in liver and muscles. Once the glycogen stored in liver and muscles are full, the remaining excess glucose is converted to lipids and stored to adipose tissues as fat. Glucose provides carbon skeleton for the synthesis of non-essential amino acid. Upon reduction, glucose forms alcohol, sorbitol. Upon oxidation, glucose forms sugar acids. Oxidation of aldehyde group at carbon number 1 produces gluconic acid. Oxidation of carboxyl group at carbon number 6 produces glucuronic acid. And oxidation of both C1 and C6 produces glucuric acid. Glucuronic acid is a detoxifying agent and takes part in the synthesis of heteropolysaccharides, hyaluronic acid, and chondroitin sulfate. Glucose combines with galactose and forms lactose, milk sugar, in mammary glands. Glucose produces NADPH2 through HMP shunt. NADPH2 keeps the glutathione of RBC reduced form and prevent them from oxidative damage. Likewise, it also keeps pentose ribose in reduced form for RNA and DNA synthesis. Glucose also produces NADH2 through glycolysis, which in turn provides energy through respiratory chain oxidative phosphorylation. Normally, glucose does not appear in urine, but when it does, then it may be a sign of diabetes mellitus. Glutathione, it is a tripeptide and is the major antioxidant produced by the cells, especially RBC, participating directly in the neutralization of free radicals and reactive oxygen compounds, hence preventing damage to important cellular components caused by reactive oxygen species such as free radicals and peroxides. Reduced glutathione serves as an electron donor. In the process, glutathione is converted to its oxidized form. Once oxidized, glutathione can be reduced back by glutathione reductase using NADPH2 as an electron donor. Glutathione is used in DNA synthesis and repair synthesis of proteins and prostaglandin, amino acid transport, and enzyme activations. Thus, every system in the body can be affected by the state of glutathione, especially the immune system, the nervous system, the gastrointestinal system, and lungs. Galactose. It is an aldohexose. It is an epimer of glucose. It is present in lactose milk sugar. It readily converts into glucose in vivo. It is used by the body for the development of brain tissues, cerebrocytes, and gangliosides. Mannose. It is an aldose sugar. It is converted to glucose in vivo. Reduction of mannose produces mannitol and alcohol, which is used therapeutically as osmotic diuretic in case of cerebral edema. Fructose. It is ketohexose. It is also called levolose due to levorotation of plain polarized light. It is the sweetest of all the sugar. The most common source of fructose is sucrose, the table sugar. Pure honey is made up of fructose only. Fructose is converted to glucose in the liver. Unlike glucose, fructose does not need insulin to enter into the cells to get metabolized. Therefore, theoretically, even diabetic can take honey in small amounts. It is present in appreciable amount in seminal fluid and acts as a source of energy for spermatozoa. Disaccharides. Unhydrolyzed disaccharides produce two monosaccharide residues. Examples are maltose, lactose, and sucrose. Disaccharides: maltose, glucose plus glucose; lactose, glucose plus lactose; sucrose, glucose plus fructose. Important disaccharides, examples and functions. Maltose, also called fruit sugar. It is also produced in the body by the actions of salivary and pancreatic amylase on starch. Maltase hydrolyzes maltose and produces two glucose molecules. The two glucose molecules are attached and with each other through a 1,4-glycoside linkage between C1 of glucose 2 molecules and glucose 2 and the C4 of glucose 1. Since we know that the glucose is an aldose sugar, it has an aldehyde group, and the aldehyde group is always at carbon number 1 of glucose, and that the aldehyde group is a carbonyl group which has reducing properties. Since the linkage is between carbon number 1 of glucose 2 and carbon number 4 of glucose 1, the C1 aldehyde group of glucose 1 is left free, which has reducing property. Hence, maltose is a reducing sugar. Lactose, also called milk sugar. Lactase, present in intestine, hydrolyzes lactose and yields glucose and galactose. Both are aldohexoses. The two monosaccharides aldohexoses, glucose and galactose, are linked together through beta-1,4-glycosidic linkage. Since the linkage between two aldohexoses is between C1 aldehyde group of galactose and C4 of glucose, the C1 aldehyde group of glucose is left free, which has reducing property. Hence, lactose is also a reducing sugar. Bacteria can also ferment lactose to lactic acid. Clinical notes: lactose intolerance, lactase deficiency. It can be caused by inherent required deficiency of the enzymes lactase. Deficient people cannot digest lactose, milk sugar found in dairy products. It is not a serious condition but may be uncomfortable. Symptoms: nausea, cramps, bloating, abdominal pain, flatulence, and diarrhea. Symptoms develop after consuming dairy products, ice cream, cake. Treatment: avoid dairy products, but since dairy products are rich in calcium, patients with lactase deficiency who avoid dairy products should take a diet other than milk, which is rich in calcium, yogurt, and green vegetables. Sucrose, also known as cane sugar, commonly known as table sugar. Sucrase, invertase, present in intestine, hydrolyzes sucrose and yields glucose and fructose. In sucrose, two monosaccharides, glucose and fructose, are linked together through alpha-1,4-glycoside linkage. C1 of glucose is joined with C2 of fructose. Since the potentially active carbonyl group, which have reducing property, aldehyde group of glucose at carbon number 1 and keto group of fructose at carbon number 2 are engaged in making glycosidic bond, there are no active groups are left free, resulting in loss of reducing property. Hence, sucrose is a non-reducing disaccharide. Sucrose exhibits the phenomena of inversions, hence also called invert sugar. Phenomena of inversions. Sucrose is dextrorotatory. It turns the plane polarized light to the right because it contains glucose dextrorotatory and fructose in its furanose form are dextrorotatory. When sucrase is added to the sucrose solution, sucrose gets hydrolyzed into glucose and fructose. After hydrolysis, fructose also changes its form from furanose to pyranose, which is very strong levorotatory and turns the plane polarized light to the left. Thus, sucrose is as a disaccharide is dextrorotatory, but after hydrolysis it becomes levorotatory. This change in the rotation of plane polarized light from right to left, from dextro to levo, is known as the phenomena of inversions. Oligosaccharides, upon hydrolysis, these sugars yield from 3 to 10 monosaccharide units, not temporary important physiologically. Polysaccharides. Upon hydrolysis, polysaccharide yields more than 10 monosaccharide units. They form structural component of cells. Unlike monosaccharides, all of the polysaccharides are non-reducing sugars. Polysaccharides are of two main types, homopolysaccharides and heteropolysaccharides. Homopolysaccharides, upon hydrolysis, homopolysaccharide yield only one type of monosaccharide units, animal source, glycogen, plant source, starch, dextrin, dextron, and cellulose. All of these contain glucose. Important hexose monophosphate HMPS examples and important homopolysaccharides examples and functions. Glycogen, it is present mainly in liver and muscles, where it acts as storage polysaccharide for glucose. Also known as animal starch. Sucrose is branched like a structure similar as to amylopectin of starch, but each branch contains less than 12 glucose residues. Amylopectin contains more than 12 glucose residues. It has two glycosidic linkage, alpha 1,4 and alpha 1,6 glycosidic linkage. Alpha 1,4 glycosidic linkage forms a straight chain of glucose in glycogen. Alpha 1,6 glycosidic linkage joins the two straight chain with each other, forming a branch. In order to release glucose from glycogen, these linkages must be broken down by two important enzymes, namely phosphorylase breaks 1,4 breaks alpha 1,4 linkage, and debranching enzyme breaks alpha 1,6 linkage. During fasting, liver glycogen maintains blood glucose level. Muscle glycogen has no role in maintaining blood glucose level. Instead, it only provides energy to the muscles. Thank you very much.